Hello, everybody. So welcome to our second lecture of genetics. And in this lecture, we will take a look at at genes, define genes, and look at the molecular basis of genes. So the term gene sprung into existence sometime in the early 20th century. And there's actually multiple people that are attributed to first using the term gene. So there was Hugo de Vries, Willem Johansen, and Bateson that basically used something like pangene, gene, or gene. And then Bateson was also the person who coined the term of our field, genetics. Mendel, right, the founding father of genetics that we will talk about a little bit later in the class, actually did not use the term gene or genetics. He talked about factors. The tricky thing, though, is even though this course is termed genetics, the way how we define a gene is actually somewhat tricky, and there is no one-size-fits-all definition of a gene. And this, despite all the advances this field has made since the early 20th century. And in fact, the definition that I'm going to offer you up is actually one that has been around since the 1920s. And I think it's attributed to Muller. So a gene under this definition is defined as, so a gene has to be replicated before it can be transmitted to the next generation. And the replication process includes the potential for variation. And a gene has to be expressed to produce a phenotype or some component of a phenotype. So basically, a gene is something that is being replicated and transmitted, and that is being expressed into a phenotype. So it's a piece of information. And you may ask, well, we know so much about molecular biology, and shouldn't we have much clearer definitions of a gene? And in fact, we will offer up a much better model for a gene a little bit later in this class. However, so much research has found that it's also other pieces of DNA, regulatory sequences outside the actual sort of general structures in the genome that we refer to as genes that also impact the phenotype. And so even though during the 1960s, 70s, we were pretty sure what a gene meant, we're now kind of going back to a more elusive definition of a gene. And of course, right, these are all in the sense we talk about gene in this class, this all relates back to DNA as the genetic material. And so we'll start with a quick refresher on DNA. So the DNA, as you've probably seen in many other courses, is a double helix, and that consists of two anti-parallel strands. So the double helix is formed out of two backbones, so phosphate backbones, one on each side. And these backbones have important consequences, right? So they are polar, as you can see by the negative charges here, and they can interact with water. So DNA is water soluble. They interact with proteins. They are very important for chromosome structure as they interact with histones and other proteins that package DNA. And this polarity is also the basis for a technique called gel electrophoresis that we'll talk about very shortly. And the two strands, are based on the base pairing rules, right? So T with A, G with C, and they are anti-parallel. So the nomenclature of the strands is basically the numbers of the carbons in the sugar, in the ribose. And the 3' sugar is up here, and here's the 5' sugar. Those are the ones that basically connect the DNA backbone, and therefore we refer to a 3' to 5' direction of the DNA. And this is just pointing out those things. And in the end, this structure forms a double helix. So this ladder will curl up on itself and form this famous structure that has become sort of emblematic for genetics. So we talked about the direction of the synthesis for nucleic acids. So if we have two nucleotides here, so consisting out of the sugar, the base, and then the at this stage as a single nucleotide, the triphosphates here. If this is the incoming DNTP as the C here, and what we call the N stands for any one of A, T, G, or C, and then this, of course, is a triphosphate. And it will actually connect to the hydroxyl group at the 3-point end, and two phosphates are produced in the process, and one phosphate basically forms the connection between the triphosphate. There was when the 5' end where the triphosphate attached, and the 3' carbon in the sugar that had the hydroxyl group that reacted with the triphosphate. And therefore, we know that DNA synthesis proceeds, and we can add another one here, proceeds in a 3' to 5' direction. So it's always the 3' ends on the burgeoning, on the newly synthesized DNA molecule that receives the nucleotides. And therefore, we have this 5 to 3' polarity. So, and of course, the basic principle behind DNA is the complementary base pairing. So and here's something you should, as a biologist, remember, even when they wake you up at 3 o'clock in the morning, completely drunk. So it is T pairs with A, so thymine with adenine, and cytosine with guanine. And this type of base pairing is incredibly important for all kinds of applications. So both natural and in the lab setting. So in nature, it is of course important for DNA replication, right? Because one strand serves as the template for the newly synthesized strand. Transcription, similar way, RNA splicing, translation, and many more. In the lab, right, this base pair complementarity is used for sequencing, hybridization experiments, PCR, gene editing, and a variety of other functions, some of which we will actually address in this lecture. So one of the main principles in sort of DNA techniques is the principle of hybridization. What we mean by that is that we have a short piece of DNA, which we refer to as an oligonucleotide, and we have a target DNA that can be much longer. It can be, in fact, an entire chromosome. And both of these are denatured, for instance, by heating them. And if the oligo finds a match on the target strand, it will actually hybridize with this target strand. And this is the principle behind a bunch of different molecular biology techniques. And it is essentially the equivalent of finding a needle in a haystack. But the thing is, right, the needle might be very small, that is the target sequence, and the haystack might be very big, that is basically our genome. But we have a probe, right, this oligonucleotide that is highly specific to the needle, and we're also flooding the haystack with a bunch of these probes. So there's a variety of applications. One of them is PCR, which we'll talk about in the future. But here's one that is really cool and fascinating. It's referred to as FISH, or fluorescence in situ hybridization. And what we can basically do with this, we can use specific probes that are specific to certain genes or chromosomal regions that have been fluorescently labeled and target chromosomes in situ, so the entire chromosome, as you see on this microscopic picture here, and find the location of genes. So in this case, for instance, it was used to, you see these red marks here, so these are red fluorescently labeled probes, and these were actually used to locate the break point of a larger chromosome, in which case the region was actually split, and we have marks on either side of this breakpoint. So it's a great way to localize genes in the actual sort of genome, and also to look for large-scale chromosomal mutations. Another DNA technique is gel electrophoresis. This is basically sort of the rite of passage for any molecular biologists, and molecular biologists are also sometimes not very flatteringly referred to as gel jockeys. So here is a DNA gel. And this is basically, DNA gel electrophoresis is based on the principle that the phosphate backbone has a negative charge, and therefore DNA molecules should be migrating in an electric field. And if we set up the electric field through a gel matrix, so in this case, this is an agarose gel, so a molecule derived from algae that is like a polysaccharide with some protein in it, it basically helps us to keep the DNA in place, but also creates, a porous surface that will actually allow smaller DNA fragments to migrate faster than larger DNA fragments. And through this technique, we can actually separate out DNA fragments of different size. So here you sort of have the realistic photographs. And this is sort of a lengthwise cross-section through this setup here. So we have a bath with buffer to basically conduct the electricity. And here is our gel. And to get the DNA into the gel, we load our sample into one of these wells. And the sample is usually mixed with glycerin or something of that sort that drags it down and sinks to the bottom, is not diffusing out. And then once the electricity is turned on, the electric field is established, and the DNA will migrate through the gel and separate according to size. So this is what you kind of see here. Samples are loaded, and here the DNA starts to migrate. And what you actually see here is not the DNA, but an indicator dye that tells you where the DNA, where DNA of particular size should be in the gel run, because, well, we'll talk about how we visualize DNA. So the DNA itself is not visible in normal light or other types of light, but we can stain DNA using a molecule called ethidium bromide. And this is a stain that intercalates. So basically it puts itself inside the DNA, and it fluoresces under UV light. And so if we expose a gel to UV light, we see those bands of DNA lining up. And what you're basically seeing here is, at the start, we have the largest DNA fragments. And as we go down each lane of the gel, the DNA fragments become smaller and smaller. And in this case, researchers use these patterns to look at genotypic differences between different strains of organisms that each show a different banding pattern. Another important DNA technique are restriction enzymes. So for instance, this banding pattern here may have actually been produced by cutting the DNA with different restriction enzymes. And what these restriction enzymes are doing, they are essentially sort of immune system responses of bacteria. So if bacteria use these restriction enzymes to find viral DNAs that have been inserted into the genome and to basically destroy them and cut them. And these enzymes have evolved to recognize specific DNA sequences. And oftentimes these DNA sequences are DNA palindromes. So what is a DNA palindrome? Well, it's a little bit different than your normal palindrome like race car. A DNA palindrome indicates a sequence that reads on the 5' to 3' direction just the same as the opposite strand in the same direction. So GAATTC also the antiparallel strand then also reads GAATTC. And this enzyme here, so Escherichia coli R1, restriction enzyme 1, recognizes this sequence and will cut the sequence, both strands at different points. So between the G, well, at the same points, but of course, these cuts are now staggered because it cuts between the G and the A in the 5' to 3' direction. And therefore, we end up with with cut strands that show these overhangs, or so-called sticky ends. And here is just another representation of it. And so different restriction enzymes, and there are different restriction enzymes that each have different recognition sequences. So I just showed you EcoR1 that has the GAATTC and then it has these staggered cuts. But this one here, SMA1, also recognizes a palindrome, GGGCCC, but it is a so-called blunt cutter, and it cuts between the G and the C on both sides and produces no overhang. And there are literally hundreds of these that are known and actually available for work in molecular labs. So as a quick repetition, just let's take a look at the central dogma of molecular biology. So we start out with DNA as the molecule that stores the genetic information. And transcription basically produces messenger RNA, and then the messenger RNA is translated into proteins or polypeptides. And those polypeptides basically form the phenotype of the organism that this gene expression is taking place in. And of course, as we heard in the last lecture, environmental factors will impact the phenotype. But there's a few other things in here. So for instance, we also have non-coding RNAs, so structural RNAs or regulatory RNAs that are not translated into proteins, but that play other roles in shaping the phenotype via gene regulation, or they might be doing things like forming ribosomal RNAs then to help with translation. And then we have, of course, DNA replication, making new DNA molecules. And this error here is reverse transcription. So this is actually something that in nature is done by retroviruses, such as HIV, that basically take their RNA genome and reverse transcribe it into genomic DNA that is then incorporated into the genomes of the host. But molecular biologists also use basically the enzymes that have been derived from viruses to help with working with RNA, because RNA is very unstable, and via reverse transcription we can turn it into complementary DNA that is much easier to work with. So in transcription, we have two strands, and one strand serves as the coding strand, and the other strand serves as the template strand. And as you can see here, the coding strand has the exact same sequence as the resulting mRNA. And we can also look at this in letter code. So, and of course, RNA has a uracil instead of thymine. But apart from that, you see that the resulting RNA has the same sequence as the coding strand. The template strand is the antiparalleled strand, and this actually serves as the template for the newly synthesized RNA strand. And so here's the same thing, just shown in a letter code. And this is important to keep in mind. So when we give the sequence of a gene, it is usually the sequence of the coding strand that is being used and reported, which makes some sense. However, when you look at the entire chromosome, which of the two strands serves as a coding, which serves as the template strand, will actually differ between genes. So here's a tiny section of the Drosophila genome. So here's the X chromosome of Drosophila. And in this spot here, we enlarged the genome, and we have two different genes here. And two of these genes, right, CG3160 and CG3149, actually use the same strand, and that is indicated by the direction of this arrow here. So if this were the, if this is the gene, well, the mRNA is basically, here would be the start codon, and the mRNA would be synthesized in this way. The gene CV, however, is encoded by the other strand. And so its start codon is here, and it's basically being read in this direction here. And also, the two genes show a tiny bit of overlap. So in this region, they actually share the same part of the chromosome, but they both read a different strand as the coding strand. So going back to the central dogma, and just as a refresher, you know that the bases of the messenger RNA are read in groups of 3. They form the codons, and each codon codes for one amino acid. And the way to read this genetic code is that, you know, the big lines here are the first letters. So all codons start with a U in RNA code here, or a T in DNA code here. These all start with a C. And then the second letter are these big columns. So here, all second letters are U's. Here, all second letters are C's. And then the third letter is that each sort of individual row is counted from U, C, A, G. And right, there are 20 amino acids that are used in natural proteins. And so we basically, you know, a 2-letter code would not be enough. We'd have only 16 combinations. So, but a 3-letter code actually has more combinations than there are amino acids. So we have 64 combinations. So 3 of those combinations are taken up by stop codons. And you know that the start codon AUG also serves as a codon for an amino acid methionine. But also we see that certain amino acids are coded by more than one codon. So those numbers can range from 6 to 1. And, you know, with only a few having 6, several having 4, others 2, and a minority of them having 3, such as isoleucine here. And you know that these codons means that therefore the bases of the mRNA are read in groups of 3. And you know, those codons are non-overlapping and therefore form a particular reading frame. And therefore, a piece of mRNA can actually be read in 3 different reading frames. So for instance, this piece of mRNA, if we start at the AGG here, we could start reading the AGG as our first codon. And if we kept going with this, this would translate into arginine, serine, aspartic acid, and serine again. Or we could start with the G. So the first codon would be GGU, and then it would read CCA. Now the sequence would turn into glycine, proline, threonine, and arginine. And if we started at the third codon, we'd end up with valine, glycine, leucine, and alanine. Of course, if we shifted the reading frame 1 base pair over, we would be basically back here. We would not read the arginine, but we would then start reading again UCC, AAC, UCG. And so this would again be serine, aspartic acid, and serine. It would then simply not include the arginine. So any stretch of DNA, right, double-stranded DNA, that's part of a chromosome, has 6 possible reading frames. And our genomes make use of all 6 of those. So here we have a unicellular eukaryote, so Plasmodium knowlesi. It's one of those organisms that are known to cause malaria. And so each of these dark gray lines is 1 strand of the chromosome. So here's 1 chromosome, and this chromosome is used as a coding strand by these genes up here. And there are 5 of them: FAT112, FAT12, FAT13, FAT14, FAT15, FAT16. And then 2 other FATs, FAT11 and FAT17, use the other strand as a coding strand. What these gray lines are, these are the 3 potential reading frames. And the way you can imagine these is that at the very beginning of the chromosome, we basically set the very start as our first reading frame. And then we jump 1 base pair over. This is the second reading frame. And then another base pair over. This is the third reading frame. And what you can see here is that, you know, these being a eukaryotic genome, well, eukaryotic genes do have introns. And so FAT17 is probably the best example for this in this graph. These have introns. The eukaryotic genes have exons. These are those boxes here. And they're connected by non-translated DNA, and those are the introns. And they're connected by these blue lines. And because introns don't have to be a length of multiples of 3, the reading frame can actually, you know, different exons can actually be of the same gene can be located on different reading frames, on different sort of chromosomal reading frames. However, when the gene is being transcribed and splicing takes place, now they all form 1 contiguous piece, and therefore the reading frame in the mRNA starts with the AUG here and is then continuous all the way through. And therefore, this is one of the reasons why errors that result in introns not being spliced out, so mutations that cause errors in the splicing, can be very fatal because they basically introduce additional DNA that doesn't really code for anything useful. Plus, it introduces a frame shift. Down here, we have a bacterial genome, and bacteria do not have, or the vast majority of bacterial genes do not have intronic sequences, as you can see here. So we don't have any sort of jumps in frame, and we don't have any of these lines connecting exons across non-coding DNA introns. So when it comes to viruses, such as our new pandemic here, the novel coronavirus genome, the SARS-CoV coronavirus 2, which causes COVID-19. These are SARS and other viruses like it, and also viruses such as measles, influenza, HIV, are RNA viruses, meaning their genome is encoded as RNA that is packaged inside the viral capsid. And these RNA viruses tend to have very short genomes, and along with it comes a very efficient packaging of the genetic information in these genomes. So, for instance, so we see things like here we have one open reading frame that is then being cut up. Once it's translated, is actually being cleaved into multiple proteins that each serve a different function for the virus. We also have like crazy things that we don't really see in eukaryotic genomes, such as ribosomal frame shifts. You kind of see that this open reading frame 1b here is slightly overlapping with open reading frame 1a. And what's actually happening is in the mRNA, well, the mRNA, there's actually in the ribosome, the mRNA, there's sort of a hiccup, and the reading frame is shifted during translation. Therefore, these genes are actually in a different reading frame as these genes here, even though they're being transcribed as part of a single RNA molecule. And then here, these genes here are transcribed as part of skipping of the RNA polymerase. So they start here at the initiation point, but then they skip over those two reading frames here and form just transcribe the S gene, or they skip over the two reading frames and the S gene and just transcribe the gene 3A and so forth. So viruses are under pressure to produce these very compact genomes, because they have to reproduce many offspring, and the genome length is actually a substantial cost to them. So the general rule that we talked about is rule of thumb is the following: that one gene equals one polypeptide. And I think this is something that has been introduced in introductory biology classes, the famous arginine synthesis pathway that was used by Biol and Tatum to explore basically this idea of one gene of one polypeptide, where we have the arginine 4, 2, and 1 genes that each are being translated into a different enzyme that serves a different function in this synthetic pathway. However, since Biol and Tatum, we learned a whole bunch more. So, for instance, one gene can result in multiple polypeptides that actually do different things, even though they are related in function and in structure. This is referred to as alternative splicing. And then some genes don't code for polypeptides at all. I mean, we already mentioned functional RNAs, such as ribosomal RNAs, small RNAs that, and so forth, that have a function without being actually turned into proteins. So the other piece of information, at least when it comes to eukaryotic genomes. And then part of this class, we will focus on eukaryotic genomes, is the modular structure of genes. So we start out here. We have the coding region of the gene, and it's actually being broken down into different exons, which are separated by introns. And of course, the coding starts here, the reading frame ATG. And this is basically what's being translated. Upstream from here, we have the core promoter that sort of is the general on-off switch of whether a gene can be transcribed. So if we use the analogy of a car, this is basically the place where you press the button or you turn the key to start the car, or you turn off the car. And then further upstream, we have the cis regulatory module. And this is basically a place where transcription factors bind and then interact with other transcription factors at the core promoter that determines where and when and how much of a gene is actually being transcribed. So this is almost like the gas pedal if we use the car analogy. And we can find, if we look at the actual sequence of a gene, we can find these elements. In this case, we'll concentrate on the coding sequence of a gene. So in what we basically have here would be like the typical structure of of a gene. So in blue colors, in blue, in green, in teal, and in hot pink here, these are exons of coding sequence. In between, in lowercase letters, in orange, are the introns, which are being spliced out. And in gray letters are the 5' up here and the 3' untranslated regions. So that untranslated regions are transcribed, so they're being turned into messenger RNA, but they're not translated. And here we have our start codon, ATG coding, and then we have a TAA as a stop codon. So let's take a closer look at the cis regulatory module. Right, so the cis regulatory module often consists out of specific DNA sequences, and these DNA sequences, they're not coding for any proteins or anything, but they serve as binding sites for transcription factors. And transcription factors are proteins that interact with DNA. They have specific domains that can recognize certain DNA sequences, such as zinc finger domains, and these transcription factors play a role in gene regulation. And depending on which transcription factors bind, this will determine how much gene is actually being expressed. And this is also the way the genome makes the different binding sites as accessible, either by unfolding them or by demethylating them, is a key element in gene regulation and helps explain why different tissues actually express different genes. So for instance, this green transcription factor the site that binds the green transcription factor might be one that is exposed in nerve cells, whereas all the other ones are basically being unexposed or silenced. And therefore, we're seeing a specific regulation of this gene in nerve cells and basically playing its specific roles in the function and development of nerve cells. So, and these are just things I just told you. And So this gene regulation of like transcription factors, so genes that are translated into transcription factors that impact the expression of other genes and can result in these complex networks and interactions of gene expression. So for instance, the transcription factor ALX um, plays a role in vertebrates in craniofacial development. So it actually is an important gene influencing the different shapes of the beaks of Darwin finches. And so in this case, it affects a bunch of genes, some of which have nothing to do with craniofacial development, but the orange ones do. And it even activates another transcription factor gene that's sort of at another level, then also activates and regulates other genes that have something to do with craniofacial development. And this is just an example of how these transcription factors form these complex regulatory networks. And every step of the central dogma offers the opportunity for gene regulation. So for instance, what we just talked about was the regulation of DNA transcription, but there are other steps. So for instance, not all RNA or not all messenger RNA is actually being translated. So it can be degraded, it can be interfered with, and it might not be actually, make it to the ribosome. And then also, right, the proteins themselves can also serve as target for regulation. So proteins can be modified after translation, and also the way they're targeted to different parts of the cell is another way of how gene regulation is being achieved. So this was a basic overview of what genes are, how they're being regulated, and the basic structure. So this should have been repetition from other courses. So when we're talking about genetic analysis, and this is what we'll be focusing on for most of this class, we're really talking about the study of genetic variation. Right? So because it is the genetic variation that leads to differences in phenotypes, and either this can be naturally occurring variation that exists in the same population or between species, or it can we can use artificially induced mutations to produce mutants in the lab that tell us something about the function of genes in generating different phenotypes. So I'd like to use one example for naturally occurring variation, and that is the example of melanism in the eastern gray squirrel. So the squirrels that you see around campus and around town, they are actually not from here. They're from the East Coast. And they normally look like this, right? So kind of gray with a little bit of white on the bottom. There is a western gray squirrel, which depends on like oak prairies, which are in savannas, which are very marginal and endangered habitats in Washington State. And hence the western gray squirrel is also quite endangered. And most squirrels that you will see are these eastern gray squirrels that are mainly right now in urban areas. What you may have noticed is that the squirrels in Bellingham come in different forms. And you might have seen some jet black melanistic forms, or sort of an intermediate looking brownish, black-brown form that is uniformly brown, but not quite as deep jet black as this squirrel here. And what's causing this this melanism in squirrels is a mutation or a set of mutations in the melanocortin 1 receptor, or MC1R. And this is from a study that was conducted on melanistic squirrels in Great Britain, where they're also an invasive species from the Americas. And what they found there was that this transmembrane region of the MC1R receptor was actually missing a bunch of amino acids, basically, um, rendering it non-functional. So what is the MC1R doing? Well, the MC1R is part of an important signaling pathway that influences what kind of pigment is being deposited in the hairs of mammals. So the pathway consists out of the gene agouti and MC1R. Agouti represses the function of MC1R. So if agouti is active, then MC1R is downregulated, meaning it has low signaling resulting in the deposition of pheomelanin, this light melanin. If agouti is not active, it will not repress MC1R, and it results in the deposition of this dark eumelanin. What happens to the mutant squirrel MC1Rs is that it basically loses its ability to be regulated by agouti. So it is a loss of function in being able to regulate it, but it's still able to put out the signals that result in the deposition of the dark eumelanin. And therefore, those jet black squirrels basically have an MC1R that is firing all the time. And we will talk about DNA polymorphism databases as part of the class discussion, but you're more than welcome to take a look at this database. I will use the I will basically stop at this point.